Okay, so tonight we're going to start talking about Chapter 2, 22, which is nucleic acids. So first of all, let's just kind of talk about what a nucleic acid is. Basically, were originally found in the nuclei of cells, and so that's why the first part of the name is nucleic for nucleus. The acid was added when the material was found to be acidic. So it was found in the nucleus and it was acidic, so they put nucleic acid together. So basically there are two types of nucleic acids. First of all, we have DNA, which is deoxyribonucleic acid, and this is found almost exclusively in the nucleus. Some in certain organelles in the mitochondria, but for the most part it's found in the nucleus. A second kind of nucleic acid is the RNA, which is ribonucleic acid, and these are found in the nucleus and in the cytoplasm. The cytoplasm is a liquid material outside the nucleus, so kind of where all of the organelles are found in a cell. There was not a huge interest in nucleic acids until 1944 because we honestly just didn't really understand them or what their importance was or why they were there. But in 1944, Oswald Avery found that he could change one strain of pneumonia bacteria into another by transferring the DNA between one cell to the other. Basically what he would do is he injected DNA from a smooth-coated bacteria with rough-coated bacteria into mice, and what he noticed is that both forms were produced. This allowed him to see that DNA was a genetic code. So basically what he did is he took a smooth-coated bacteria and he injected it with rough-coated DNA. And so when he put this rough-coated bacteria in the mice, rough-coated bacteria was not the only thing created. Both rough and smooth-coated were created, even though he had only put the rough-coated or the smooth-coated in the mice. This was evidence that one species varied from another. So let's start talking about the structure of DNA and RNA. Polymers are made from three different compounds. So when we're talking about DNA and RNA, there are three different parts. We have the sugar, we have a phosphate group, and we have 4 nucleotide bases. So if we look at this chart, it shows the differences between the DNA and the RNA. So the DNA contains the sugar deoxyribose, it contains a phosphate group, and it contains 4 bases: adenine, guanine, cytosine, and thymine. RNA contains ribose sugar, it also contains a phosphase, and it contains adenine, guanine, cytosine, but instead of thymine, it uses uracil. So the DNA and RNA are very, very, very similar, except for DNA has deoxyribose and RNA uses uracil. So there's really only 2 big differences. The nucleotide bases are usually just represented by their first letter, and so we use A for adenine, T for thymine, U for uracil, G for guanine, C for cytosine, and then you can see here that you can see the pictures of the ribose and the deoxyribose. So if we compare these, we can see that they're pretty close except for right here. Ribose has an extra hydroxide group added to it that deoxyribose does not. So the nucleotide bases are very important because this is what makes your DNA. This is the code. This is the directions for how your cells work and how your body works. Well, in 1950, Erwin Chargaff came up with Chargaff's rule, and basically what he discovered was the amount of adenine is always equal to the amount of thymine, and the amount of guanine is always equal to the amount of cytosine. And so based on that, he was able to kind of put together that A must always bind with T and G must always bind with C. In 1953, James Watson and Francis Crick, which I'm sure you've heard of them before, based on the X-ray diffraction by Rosalind Franklin and Maurice Wilkins, they were able to propose that DNA had a double helix structure, which is kind of like a twisting spiral staircase, and they actually won the Nobel Prize in 1962 for their work. So this is what the double helix model looks like. As we can see over to the side, it looks like a twisting staircase. And if you look, adenine, which is yellow, and thymine, which is green, are always bound together. So one strand will have the A, the other strand will have the matching T. And same thing with cytosine and guanine. This model suggests that there are 2 backbones with alternating sugar and phosphate groups. So basically what happens is it goes deoxyribose, phosphate, deoxyribose phosphate, deoxyribose phosphate, all the way down the sugar backbone, which are the red and the green strands that you can see in this picture over here. He said that they are helically coiled like a spiral staircase, and the bases are attached to the sugars, and they kind of act like steps on the staircase. So like I mentioned earlier, A always goes with T and C always goes with G, and these are held together with hydrogen bonds. Now hydrogen bonds are kind of a special case of bonds, and we didn't really talk about them a whole lot when we were talking about bonding a few chapters ago. But hydrogen bonds are not ionic or covalent. Just to give us a little bit of background, sorry, I forgot to take that off. To give a little bit of reminder about what ionic versus covalent is, ionic is when we are dealing with charges, and these deal with the loss of electrons. Covalent deals with sharing electrons. Just to kind of review really quick what that means. So if they're not ionic, which means they don't lose or gain electrons, and they're not covalent, which means they're not sharing electrons, well then they have to be doing something, because a bond has to be doing something with electrons. They have to be attracted to one another in some way, shape, or form. And so basically what a hydrogen bond is there's an attraction between the slightly positive hydrogens of one base and the slightly negative nitrogen oxygens of another. So if we look at adenine and thymine, okay, we have a hydrogen right here, which is just a little bit positive, because right here is a covalent bond. Nitrogen is stronger than hydrogen, so he's going to be in control of the electrons. So therefore, hydrogen is going to be slightly positive. Over here, there's a covalent bond right here between oxygen and carbon. And since oxygen is stronger than carbon, he's going to be in control of the electrons, so that makes him slightly negative. And so therefore, there is a attraction between the slight negative and slight positive, just kind of like there's an attraction between the north and the south pole of magnets. Same thing occurs over here. We have a slight positive charge on this hydrogen, a slight negative charge on this nitrogen. So again, there is a attraction there, and that is what holds these together. Now, it's a weak attraction, so that bond can be easily broken, but at the same time, that's what's holding them together. So if we look at a bigger picture of what's going on, we can see not only can we see the hydrogen bonds in this picture, but we can also see that in thymine and adenine, there's only 2 spots for hydrogen bonds to form, so that's why they always bond together. But if we look at cytosine versus guanine, we can see that there are 3 spots for hydrogen bonds to form. So that is why those are always bonding together. Alright, so now let's spend a little bit of time just talking about DNA. So DNA is located specifically in the nucleus. Again, sometimes it's found in ribosomes, in some other organelles in your cytoplasm, but for the most part, it's only found in the nucleus. DNA can be organized in genes, and genes spell out the traits of your body. So they give a way to differentiate between one species to the other. They are also strung together in something called chromosomes. Humans have 46 chromosomes, which is 23 pairs. You get 1 pair from the male or your father, and you get 1 pair from the female, your mother. DNA, in order to go from cell to cell, it is divided, and then it is replicated to reproduce identical cells. So this is how your body reproduces cells to fix damage or maintain life, so on and so forth. So let's talk about this replication process. So in replication, the double helix must be divided into a left-handed strand and a right-handed strand. The left-handed strand can then make a matching right-handed strand, and the right-handed strand can then make a left-handed strand. So it's kind of like if you have a puzzle and it has a cutout, or if you're making cookies and you use a cookie cutter and you have a star cookie cutter, if you go to make a star cookie, you don't get a new star cookie cutter, you get a cookie when you use the cookie cutter. And so it's kind of, they make the opposite of what they are trying to make. So, side fact about DNA, it is very, very, very coiled. DNA is very long. It would be approximately 6 feet long if we unwound it and stretched it from end to end. And so in order for it to fit into the nucleus of a cell, it has to be coiled up very, very, very tightly, kind of like a telephone, those old telephone cords. And not only that, but in the replication process, if we tried to unwind the entire piece of DNA before we even started to replicate it, it would take way too long because the replication process needs to happen relatively quickly because your body is needing to reproduce the DNA on a much faster basis. So in order to get this process to speed up, we actually use enzymes because if you remember, enzymes speed up chemical reactions. So the enzymes that are mainly involved in this DNA replication, so if we look at this picture, we can see that this is DNA, and it is coiled up kind of like a telephone cord like I was telling you about. And basically what happens is we have an enzyme, endonuclease, and basically what it does is it chops up the DNA. So it separates the right-handed strand from the left-handed strand. And then we have another enzyme called polymerase, which is the yellow thing right here, that kind of goes along the side. I feel like it's kind of like a zipper, kind of goes along the half DNA strand. And what it does is it copies the DNA, and then we have the enzyme ligase, which honestly kind of sews it all back together. And so in the ending, we get a new set of DNA. It's half old and half new. Well, DNA in itself is just a set of directions. What DNA does is it tells your body how to produce proteins. So proteins are the main structural material in the body. So for instance, we talked about these a few chapters ago. We talked about collagen and keratin. Basically, these contain enzymes, and the function is determined by their amino acid sequence. So how does the sequence of bases in DNA lead to the sequence of amino acids in the proteins? Honestly, it's transcription and translation. Like I said, DNA is just a set of directions. It's how we use the directions to make the proteins that are necessary for life. And so, first of all, we have transcription. Transcription takes place in the nucleus, and what happens in transcription is DNA is copied into RNA. Because DNA doesn't really usually leave the DNA, the RNA, therefore, takes the directions outside. They're kind of like the contractor on a job site. The contractor goes and talks to the people that they're building the home or the business for, and then he brings the message back to the workers and makes sure the workers do what they're supposed to be doing. So that's kind of what the RNA is doing. It takes the directions and it goes and tells the body what to do. The RNA is very similar to DNA, but it has a few differences. So basically, it exists as a single strand, so it doesn't have matching base pairs. It exists as one single stair, so kind of like half of a staircase. And the backbone is made out of sugar called ribose as opposed to the deoxyribose, and it uses uracil in place of thymine. So, which sounds like a huge deal, but when we actually compare uracil and thymine, they look really close to each other, and the only difference in thymine and uracil is this group, this methyl group right there. Uracil doesn't have it, whereas thymine does. So here's kind of a picture to help you see the difference between DNA and RNA. DNA, again, is a single strand. It still looks like a spiral staircase, but it would be kind of hard to climb this because you'd only have half a step and one handrail as opposed to DNA, which is a true spiral staircase. Okay, so basically what happens in transcription is the RNA is built to pair with a strand of DNA. So if this is our strand of DNA, and remember it's a set of directions telling your body what proteins to make where, this is what you would see. And so basically what's going to happen is we are going to break this, and the RNA is going to match up that top strand. So C is going to make a G, T is an A, C is a G. This time A is going to give you a U. So if you notice, this strand and this strand are identical, except where you see a T on the top, you'll see a U on the bottom. And so basically this strand is complementing this strand, which means it's kind of like the mirror image. The RNA, once it is created, then leaves the nucleus and enters the cytoplasm, and it begins the next process called translation. Again, RNA in this case is serving to take the directions from the DNA out into the cell to tell them how to make the proteins and what the purpose is. So after transcription, so after we make the RNA, we move on to translation. Translation takes place in the cytoplasm, and basically this is where your proteins are assembled. In order for your proteins to be assembled, we actually use ribosomes, and these are often called protein factories. because a protein has to be assembled kind of like in an assembly line at a factory. And so in order to understand this process, we need to talk about the 3 different types of RNA. So first of all, there is ribosomal RNA, which is called rRNA, and this is kind of like the factory. This is where the protein assembly takes place. The second kind is called the messenger RNA, or the mRNA, and this is honestly like the blueprint. And so it's like the RNA from the previous transcription step. It's taking the message from the DNA into the cell. That is your messenger RNA. And this contains the code for the sequence of the amino acids. So it's going to tell the ribosomal RNA how to make the protein. And then we have transfer RNA, tRNA, and these are kind of like the delivery trucks. They run around the cell and they find the materials that you need in order to create the protein. So just to kind of refresh, we have, here's our nucleus, and in our nucleus we create our mRNA. Our mRNA then comes out here, and it goes over here to our rRNA, our ribosomal RNA, or the factory, and it is telling the factory how to assemble our amino acid chain right there. And these guys right here are your tRNAs, and basically what they do is they go out and they find the amino acids that you need. They bring them to the ribosome in order for the ribosome to match up with the code on the RNA to produce a longer and longer amino acid chain. So what does all this mean? Well, this basically leads us up to something to the genetic code, and basically in our body we utilize 20 amino acids. Each amino acid is coded with a triplet, and a triplet is just basically a series of 3 amino acids. So since there are 4 bases and we all use triplets, it should theoretically allow for 64 amino acids, which is way more than the 20 I just told you that we use. So therefore, each amino acid has a different triplet code. So it would be like having more than one phone number. If you're trying to get a hold of me, you could call my cell phone number. If that doesn't work, you could call my work phone number. If that doesn't work, you could call my home phone number. If that doesn't work, you could call my husband's phone number. And if you still can't find me, you could probably give my mom a shot. But it's just, you can work down the line. There's lots of different ways to get a hold of somebody, and so it's kind of the same way. A lot of these guys have more than one code, and it will code for the exact same amino acid. So like for example, glycine, which is shorthand GLY, has the code GGU, so guanine, guanine, uracil, GGC, guanine, guanine, cytosine, GGA, guanine, guanine, adenine, and then GGG, which is just guanine, guanine, guanine. So when these amino acids, or when those bases all line up in this specific order, it'll tell the body that it needs to produce glycine at that point in your amino acid. And the good thing about genetic codes is they are universal, which means they work in bacteria, plants, and animals. So it doesn't matter if we're talking about the DNA from a flower that we found outside or the DNA from your pet. The codes are the same, the amino acids are the same, and so it makes it really easy for us to study DNA from various species. And so just to kind of give you a look, here are our 20 amino acids that we use and their abbreviations. And if you look, some of them, the majority of them, have more than one code or phone number in order to tell them. There's a few down here that only have one, like methionine and tryptophan, but the rest of them have more than one triplet in order to read in a RNA or DNA cell. Okay, so let's talk about those codes a little bit. Not all codes are for amino acids. So when I say a code, I'm talking about those triplets that I just showed you the chart for that tell you what amino acid to put in what sequence in the protein chain. And so, but not all of them are telling you for amino acids. Some of them act like punctuation marks. So whenever you see UAA or UAG or UGA, and again, these are your bases on your RNA, this is telling the ribosome to stop. It basically is saying that your protein chain is complete and you need to stop making your amino acid sequence and you need to release the protein to go do its job now. When we're doing about this, these codes also have a few other names, and they can be called the codon. This is a 3-letter code on the mRNA versus the anticodon, versus the anticodon, which is a 3-letter code on the tRNA that pairs up with the codon. So if you look down here at this picture, we have the mRNA, which is telling us what protein to put or what amino acid to put in what sequence on our protein. And if you look, the tRNA is the exact opposite. So it's like they're pairing up, just like the DNA pairs up. And so the bottom, the mRNA that comes from the DNA is called the codon, and the tRNA is the opposite of the codon, which is why we call it the anticodon. So let's spend a little bit of time talking about mutations. Now, mistakes can happen. This is a very specific process, and mistakes can happen along the way. They don't happen very often, but they do happen. If a mutation happens in translation, and remember, translation is when we are making the messenger RNA and the nucleus from the DNA. So if our messenger RNA is made incorrectly, it's not a huge deal. Basically, those proteins are not going to work properly. And it's kind of like comparing it, if you are a factory worker and you're working in a factory and you're assembling thousands of toys or whatever you're assembling every day. It'd be like you making one toy incorrectly, but the rest of the day maybe you make 500 toys correctly. And so maybe there'd be one protein in your cell that wasn't 100% correct, but the rest of them would work and that would be just fine, so it's not a huge deal. If a mutation occurs in transcription, remember transcription is when you are making your amino acid sequences, the blueprint would have a mistake as well. So every protein you made would be wrong, every single one of them, because the directions are wrong. If I give you a test and I give you incorrect directions, then everybody's going to be doing that part of the test wrong just because I gave you the directions wrong, not because you didn't understand it. And so that's a little bit more of a serious. If your blueprint's wrong, then everything you make is going to be wrong versus just one aspect of it being wrong out of 500. However, if mutation occurs in replication, it is a very, very, very big deal. Remember, replication is when we are recreating our DNA for when our cells duplicate. And basically, if our cells contain incorrect DNA, which make incorrect RNA, therefore we would then make incorrect protein. So it'd be like everything, our set of directions is wrong, our blueprint is wrong, we're making incorrect proteins, and so honestly, everything we're doing is wrong. In most cases, the cell dies because the directions are wrong, and so it can't survive, and so it dies, which is no big deal. However, sometimes a cell will continue to multiply, and this is what causes a tumor. If the tumor then spreads or metastasizes, this is when we call it cancer. So when it's spreading from one part of the body to the next, we can call this cancer. If this occurs in a sex cell, so like in your egg or sperm, the mistake will affect every cell of the human being. And so basically what this is it will relate to a genetic disease. But since we have 23 pairs, the hope is that you're not going to get a bad gene from both your mom and your dad. So that is why genetic diseases are rare, is because you have to get a bad gene from both your mom and your dad, and you have to get the same bad gene. And so the chances of that happening are pretty slim, but it does happen. So one case of it happening is sickle cell anemia. Basically in this, there is one little bitty mistake in your DNA. There is a single wrong amino acid in the hemoglobin protein. So when your body is making hemoglobin, it is making it just a little bit wrong. It still works, it's still hemoglobin, it's still transporting oxygen. It just has a little bit of a shape. So instead of the code being CTC for glutamic acid, it is changed to CAC, which is valine, which again isn't a huge deal. You still have hemoglobin. However, when your oxygen levels are low, these proteins tend to clump up and it causes a sickle type shape in your red blood cells. And then your red blood cells can't fit through the capillaries, which deprives cells from oxygen. And this can cause a lot of issues, specifically in your spleen. A lot of people with sickle cell anemia usually have to get their spleen removed because the spleen recognizes these as defective and it destroys them. And then at the same time, it's being deprived from oxygen. And so if the wrong part of your body is deprived from oxygen for too long, then it can cause some really big issues. But the good part about some genetic diseases is sometimes they might be bad in one case, but they are good in another case. And so genes usually come in pairs. And so when we're talking about pairs, we use capital letters to represent the normal genes, and we use lowercase letters to represent the defective gene. So if we have a normal gene from your mom and a normal gene from your dad, we use 2 capital letters. And this means that you're going to have the normal gene all the way around. You're not going to have any sort of mistaken protein in there at all. If you're given little h, this means that you will have the genetic disease of whatever that particular gene will cause. But if you have one good gene and one bad gene, this will make you a carrier. And what that means is you will not have the symptoms of the disease. However, you could pass it down to your children. So if you marry somebody who also is a carrier for this disease, you could have a 25% chance of passing this on and having a child with this genetic disease. So this is how the genetic diseases still kind of play out. But in order for you to have it, both parents have to have the gene in order for this disorder to be present. So if this is the case, why do the diseases prevail? Well, sometimes, like I said a minute ago, they can be a good thing. So if you have sickle cell anemia, which is prevalent in African Americans, it also can make you less susceptible to malaria. So if you have 2 bad genes, one from your mom, one from your dad, you usually die really young because of the complications that come with it. If you have capital H, which means you have 2 normal genes, you could have a higher chance of dying from malaria. However, if you have one good gene and one bad gene, there's something in the shape of those red blood cells that malaria is a lot harder to take over the body and take over that, and it's a lot harder for you to become sick from malaria, and so you have a better chance of survival. So this is how this gene stays in our population, because sometimes it gives you an added benefit. Okay, so some more genetic diseases would be cystic fibrosis, and this is, again, just one amino acid is missing, and this causes your lungs to clog with mucus, which makes it hard for you to breathe. Another one is Tay-Sachs disease. This is a buildup of fats in the brain, which can cause brain damage. And then we have phytoketonuria, which basically this is a defect in the enzyme that breaks down the phenylalanine to the tyrosine, and basically it just converts it wrong. What it does is but when it converts it wrong, it converts it into something that is very toxic to your body, and then it can lead to brain damage. And so oftentimes, if this is caught early enough, they can give you medication to break it down in the correct way. But a lot of times in babies, it takes a long time for them to figure out what's wrong with the baby, and so by the time they figured out what's happened, usually brain damage has occurred. And so this is one that's harder for them to diagnose. So this leads us to genetic engineering. If we know about genes, we know good genes versus bad genes. Is there ways that we can add good genes to things that maybe we need them, like crops? We need them to grow faster and longer. Okay, so in genetic engineering, we can have a transfer of genes between organisms. And so basically these can be called recombinant DNA. And in this, we can do transgenic. And this is when an organism contains genes from another organism. And we also can use viruses. So if we have an organism that contains genes from another organism, how in the world do we get those genes into the other organisms? And oftentimes we use viruses. Viruses by themselves are not harmful because they are nothing more than just DNA and RNA, which remember is a set of directions. And so, but they're kind of like a Trojan horse. They're not bad in themselves, but if they get into your body and they start attacking your cells, basically what they do is they invade a cell, it takes over and causes the cell to then produce more viruses as opposed to what the cell was initially intended for. So that cell will stop making the proteins or whatever it was supposed to be making, and then it starts to make viruses. Well, after some point, the cell can no longer contain the viruses, and then the cell bursts, and all the viruses are released, and then those go and attack other. So that is why oftentimes when you have a virus, it takes a few days for you to start to feel the symptoms, is because it takes a while for the viruses to attack enough cells for you to feel the systems. This can lead us to a genetically modified, and this is a big push in plant crops. And so we have the flavor saver tomato, and basically we have genetically modified them to ripen more slowly. We also have the BT potatoes. Basically what these potatoes do is they produce a toxin that kills insects, and so it's like an insecticide without having to use one. And we actually have made corn, cotton, canola, sugar beets, wheat, and alfalfa that all can kill insects just because they have a modified gene. And then, of course, we have the Roundup Ready, and basically these are soybeans and cotton that you can use Roundup on, so we can use an insecticide or herbicide to kill all of the weeds and other plants, but at the same time it doesn't kill those specific type of plants, so that's good. Crops can actually be modified to improve nutrition, and so there's a lot of vitamin A deficiency in Africa and China due to the diets and the foods that they eat. And so if we add vitamin A in rice, which vitamin A can be taken to prevent blindness, well, they eat lots of rice in China and in Africa because it's cheap and it's easy to fix and there's lots of it. So if we add vitamin A to it, then it should reduce the amount of blindness, and we actually have seen a much better rise in, or I guess a decrease in the amount of blindness in those areas of the world. These can actually also occur in animals, and in 1999 they were able to make goats that could excrete spider web protein in their milk. I'm not exactly sure why we would need to do that, but maybe down the line that could be beneficial for some way, shape, or form. Another example is salmon. We have genetically modified salmon, and basically these salmon can grow twice as fast. So this is really helpful when we're trying to harvest salmon for food. Another thing is DNA fingerprinting. So when we're talking about identifying somebody based on their DNA, we use the non-coding regions of DNA because these are very, very unique to an individual. The majority of our DNA is identical because we're all humans. However, we have to look at these non-coding regions in order to see individual differences, and these are your blood, your hair, and your saliva. These can be used to identify a criminal or to test for paternity. They can be used to identify, we use them to identify one suspect in the World Trade Center bombing, and basically what happened is the suspect licked a stamp he sent to the FBI, and from that we were able to backtrace that it was him who sent the original letter. This was also used to free several prisoners that were once thought to be rapists because they had collected the DNA and everything from the rape scene, but they didn't know how to test it. So once the technology caught up, they were able to go back and test that, and they were actually able to free some of these prisoners because they weren't guilty. And we actually recently used this to confirm that Easton Hemmings, which was the son to Sally Hemings, which is one of Thomas Jefferson's slaves, that we were able to prove that he was indeed the son of Thomas Jefferson, which we had always kind of alluded to because he had red hair much like Thomas Jefferson, and that is very unusual for slaves at that time. Two more things we're going to talk about. The first one is the Human Genome Project, and basically we're just trying to map the human genome. We contain 21,000 genes, which is far less than they were expecting. They thought that they were going to map 100,000 genes, but only 21,000, which is the same as a chicken, and half of our genes are the same as a banana. And so we are half the same as a banana, if you want to think of it that way. Evolution and the origin of life. Some mutations are beneficial. Not all mutations are negative. Some of them help you to survive better. Just like I was telling you about the sickle cell anemia. That helps people in Africa fight malaria, and so that is why it is prevalent. It is actually a beneficial mutation, and so that is why it survives. Beneficial mutations survive, and this because they allow the processor to thrive and reproduce down the line. If they are able to reproduce, then those genes get passed on to the next generation. Bad ones usually die off because it decreases the possessor the ability to survive and reproduce. If they die at a young age, they're not going to be around to reproduce, and so therefore that gene does not get carried on down into the gene pool. However, some of them are neutral and play no part in the survival, and so this is kind of like hair color or eye color. It doesn't really matter. It's not going to affect your ability to survive, and so those don't really die out or be preserved because they're just kind of there. They're not really a huge player.